Ladies and gentlemen, welcome to this latest teaching session in the course on technology and the future of medicine. Today I'm speaking about an introduction to regenerative medicine. And maybe the most logical first question is, well, what does that term mean and how is it different from the others that I've heard in the area? So there is also tissue engineering and there is also cell therapy. And this picture gives you an idea what the trends are. Cell therapy is apparently in 2016 much sexier than the other terms and much more likely to be used. It's a very vague term. I mean, cell therapy could be virtually anything. But the use of tissue engineering has been trending down, and regenerative medicine has stayed just about the same over the past umpteen years. So basically we're talking about stem cell creation of organs, stem cell repair of organs, and the idea of the end of the organ shortage. In current medical care, we are providing transplants to only 10% of the people who need them, so 90% of people who need a replacement organ don't get one. And you can imagine in the era of regenerative medicine that there will be no shortage. Everybody who needs an organ will be able to receive one. And then there's the exciting prospect that the organs provided won't be quite normal because embryogenesis in the womb over 9 months is completely different from the way these organs would be created. So they might have things missing, they might have extra things, they might create tumors, they might not function in a normal fashion. So anyway, these are some of the considerations around regenerative medicine. These are the actual numbers for 2014. This is the latest data about transplantation worldwide and the number of different kinds of organs. You can see that the kidney, which is a paired organ, many more of those are transplanted than the other single organs: liver, heart, lung, pancreas, small bowel. But overall, we reach less than 10% of the global need for replacement organs. So thinking about it simply, worldwide 1.2 million people are in need of transplantation for end-stage organ failure each year, and current transplant protocols reach fewer than 10% of them. So regenerative medicine has the potential of saving the remaining 90%, that's over 1 million people annually. So that's impressive, right? So in a sense, regenerative medicine is already here. For simple tubular organs, it's fairly easy to make organs from stem cells. So bladder, trachea, esophagus, vagina. Those of you who are familiar with this lectures I've given it before will realize there's a slide missing already, even though we're only on the 5th slide, and that's the trachea slide. Now, why did I miss out the trachea slide? Well, that also gets to what my general attitude toward regenerative medicine is. It'd be simpler to teach you guys if I had just one view. Like, regenerative medicine is great and is the future of medicine, full stop, period. Or it's a hoax, or it's overhyped. And so the fact is that if you look at intellectual dishonesty in science, it is greater in this area of medicine than in any other. If you look at inappropriate media hype of expectations, it is greater in this area than in any other. And it's also true that regenerative medicine does hold greater hope for improvement of the lives and health of human beings than any other area of medicine that we know of. So as I'll talk about in a few slides from now, this is sort of like the idea of double think or triple think in the book 1984, where you take two conflicting ideas and accept them both, keep them both in your mind. You almost have to do that for this. So, why is the trachea missing? The trachea is missing because there's one sort of evangelical character who is associated with all the trachea work thus far, and he's been found to be completely dishonest. Most of his patients have died, and it's a major 2016 scandal that the ultimate outcome of these regenerative medicine created tracheas has been very, very bad. And a lot of prestigious institutions have been associated with this trachea scandal. So when I found out about this, I rushed in and removed the trachea picture because I don't want to be supporting this charlatan. But on the other hand, every year there's more positive results and good stuff happening, and no matter what you do with your life, it will impact you soon. There will be, diseases that were untreatable before that are now readily treatable through the use of stem cells. It'll be wonderful, but there's also a quite terrible side of the evil that human beings do, and how that sometimes is opportunistically, specifically applied to the most promising areas of medicine. You're in a very good place if you want to follow what's going on in stem cell replacement of organs. Dr. James Shapiro, who I've worked with closely over a long period, it is the lead investigator in the ViaCyte study, which uses a sort of sealed capsule that fluid can get in, but cells can't get in and out of pancreatic progenitor cells as a treatment for type 1 diabetes, and thus far this trial is looking very, very good. On the other hand, the greatest expert on the hyping and the inappropriate ethics in this area is also here. So Timothy Caulfield, who's one of the most popular speakers on campus, and probably most of you know him because of that, and he's as close to a celebrity speaker as we have. He knows many celebrities personally. He's studied them. He's written books about them, like Is Gwyneth Paltrow Wrong About Everything? Is his latest book. But this is the area that he's focusing most on. So, and it's a really big area. Stem cell tourism is estimated to be a $3 billion a year industry and growing. And there are more than 700 stem cell therapy clinics worldwide, most of which are giving completely undocumented care with no proof of any benefit of the care that they are providing. But the celebrity side of this is very strong, and the sports side. And none of you are sort of immune from that. Probably all of you have a favorite celebrity and maybe a favorite sports star, and you're completely logical, intelligent human beings. But if your favorite sports star or celebrity goes to Mexico or China for some miraculous stem cell cure and comes back claiming it's the best thing ever, you will be impressed by that. And you will be likely to follow in that person's footsteps when you need some health intervention. Now, in the next few slides, yes?
So, is it therapy provided as experimental, or is it like a legitimate clinical therapy in those clinics that do
I would say that it is presented as if there was already proof that this works, and therefore come and benefit from this stuff already proven to work. But in fact, there hardly ever is published literature evidence. So in that sense, those statements that it works are always false, in the sense that any legitimate hospital or clinic that was doing research that had favorable outcomes would publish that. So there would be a paper showing that this works and that what you're going to get done, you know, it is actually legitimate. And that is almost always never the case. So in the next few slides, Timothy Caulfield is amazingly productive. He's not just a good speaker, he spends some part of the day writing. And I just want to, whatever your personal interests are, some of these articles that he's penned in the last 4 years are going to interest you. Science, celebrities, and public engagement, science confronting stem cell hype. This is mainly about the fact that every article in the lay literature about stem cells as a therapy almost always promises it will help more people sooner than it possibly can. So it's not that the promise that they hold out is absolutely false. Maybe it can do the things that they are suggesting, but not as quickly and not for as many people. Media portrayal of non-invasive prenatal testing, marginally scientific genetic testing of children and adolescents for lifestyle and health promotion, trafficking in human beings for the purpose of organ removal, ethical, legal obligations. Some of these things are bound to interest you, even if like your ethical sensitivity is extremely low and you don't really give a darn about human ethics generally. Some of these things will still turn your crank. Stem cell hype, media portrayal, therapy, policy options, athletes, and unproven stem cell therapies. You'd be amazed at the number of athletes who have had such treatments and how prominent they are. Research ethics and stem cells, is it time to rethink current approaches to oversight? So anyway, you get the idea, and there is nobody like him anywhere else as far as I know, so you're at the place that's sort of the center of looking at stem cell hype. Okay. And the other potential positive aspect of stem cell therapy is it has the potential to reverse three looming problems in medicine, and particularly in areas of medicine that interest me. Those areas of medicine are getting a little bit dull. There are fewer, the best and brightest young people going into those areas, and more people are leaving, and salaries are going down, and support from industry is going down. And so there is what they've been calling a loss of luster. 30 years ago, when you talked to a transplant surgeon, it was kind of ridiculous when they talked about themselves and the kind of person they are. Say, you know, if you go to a party, you can tell the transplant surgeons they have that extra special spring in their step, and like they're these magical human beings. And now it's come down to the fact that it seems today in 2016 like transplant surgeons are just sort of regular doctors, nothing particularly exciting or special about them. So there's been a tremendous loss of prestige and luster, and young people notice that. So like people who think themselves maybe have this special spring in their step and they're special people are less likely to go into transplantation and kidney medicine than they once were. So I used to kid that when robots replace everybody else, that nephrologists and kidney pathologists will still be employed because the problems we face are so intellectually challenging that robots will never be able to figure out what we do. Of course, that's entirely false. But some of the things I'm talking about today are a little bit complicated, and at least for a while, they'll probably puzzle sentient machines just like they puzzle human beings right now today. Now, as you may know, I'm responsible for the Banff classification of kidney transplant pathology. In medicine, there are a lot of classifications. There are classifications of this, that, and the other thing. And what is remarkable is not that I created a classification, because I think anybody could do that, and a lot of people have. It's the fact that it's the only one. How did I do that? Like, did I murder all my competitors? No, when points were reached where there was a voice from the outside, raising additional points or thinking we should modify things, I tended to embrace those voices, bring them in, make them part of what we were doing. So by continually being flexible in what the Banff classification is and wanted to be, I've been able to make it the worldwide standard. So it is the only game in town, and it's not like it was in the beginning, because I've compromised at multiple points to bring in good ideas from other people. So this happens to be the 25th year of this Banff transplant pathology consensus process that I lead. And so it's interesting, what do young people think about this? It's one thing to hear an old guy talk about it who's been doing it for 25 years, and I was kind of amused with this idea. Do you know what imprimatur means? So in the old days of printing presses and so on, you needed to get authorization to print something. And so that's what imprimatur means. Let it be printed. And now the lesion scoring, the looking at individual kinds of changes in tubules and blood vessels and the space between the two tubules, we have all these various scores. You see G, CG, I, CI. Boy, it gets boring pretty quickly, eh? But what young people say is being able to do that lesion scoring proves that you're an educated kidney and transplant pathologist. And I never really thought about it that way before. I'm not sure it's true, but I just thought that apparently is the way young people look at this. Once you can talk in Banff lesion scoring terms, then you're worthy of respect in the field. So these are the milestones of what we've done. So it's not just kidney, it's all solid organs. And it's if you want to think about things that you yourself might do, it's kind of intriguing how wrong I was about this in the beginning. In 1990, the heart people created a heart transplant pathology classification. And I thought I was just copying them. I mean, in essence, there was nothing creative. It was like a knee-jerk thing, right? So they did it, and I was going to do it. But they had pathologist authors only. They had no clinicians, no surgeons, no scientists, no ethicists, no people from the outside. They had only pathologists. And there's been endless bickering and all sorts of political problems because of the way the thing was structured. Whereas I started out naively, because I didn't know of any better way to do it, of having all the stakeholders represented from the beginning. And that turned out to be a very good idea. I also didn't really trigger to the idea that you can make any money by creating classification. I regarded this as a purely academic exercise, no financial implications. And that also turned out to be false. So I'm happy I was wrong about all those things. But if you yourself start something, it just shows you that you can make a success of something, whereas your initial understanding was so seriously flawed. If you thought that, you have no business leading this. But anyway, that's what I thought. It was a purely academic exercise, and we were just copying the Heart people. But ultimately, that isn't so. We're now regarded as the leaders of this, and surprisingly, for something that's 25 years old, it has more activity in terms of published papers about it now than in any other previous year, and you can see that it's steadily going up. And I'm going to talk later about tissue engineering pathology, which will probably also increase the number of papers. Now, you may think, well, I must have given this talk a whole bunch of times before, and this must be very similar to previous times. Wrong. So you see that 2005 peak. This has always bothered me. And last night, as I was creating this slide set for you, I thought, I'm going to fix this for once and for all at tomorrow's class. So every other audience who's heard me talk about this has been left completely mystified by that 2005 peak. I've talked about a kind of combination of different poorly understood effects that led to that 2005 peak. But today, for you only, I'm going to reveal what I believe the true cause. It will astound and amaze you. So, in November of 2005, as some of you know, I went to visit Leonard Cohen for a 3-day weekend. Now, my fear was this famous, iconic poet and singer, songwriter would be completely bored by me, and the social situation would be wrecked by what an extremely boring person I am and how exciting and, intrinsically creative he is. And the whole thing would just fall on its face because of my lack of stellar characteristics for that kind of meeting. And boy, was I wrong. So he was much more interested in me than I was in him. And so these 3 days, he was interrogating me about what it's like to be a doctor, what standard setting is like, what it's like to read slides under the microscope, all sorts of things that I thought no other human being would ever be interested about my life. Leonard Cohen was actually interested. And we talked about how to use medical means to determine where the Leonard Cohen fans were, and we talked about maybe there's something uniquely in the water that one could detect, because that was a big problem for him. You had these gigantic cities, and you knew that there were a substantial number of Leonard Cohen fans in these big cities, but you could spend decades and not find out where they are and who they are. So it was really interesting, and of course, as I've told you, the Sony Aibo robotic dog, which you met earlier this term, Leonard also met. Now, what is the evidence that 3 days had any impact on him? Well, the next year, he won the Lifetime Songwriter Award, and in his acceptance speech, he talks about living life with microscopic precision. How about that, okay? So that's what I'm claiming. It's as good as any other way to explain that peak that I can think of. It brings in the arts, so Earl will be very pleased with that. So it kind of embraces all considerations. And so now the 9 of you here, the only human beings who know the reason for this 2005 peak in the Banff classification papers. Okay, there it is in all its glory. The way the meetings work is sort of unique in that a lot of the meeting is free-flowing consensus generation rather than just having one boring paper after another at set times. We set aside long blocks of time for generating consensus. And that's illegal. Did you realize that? But if one of you were to set about to do consensus generation, and if you were to look up how to do it, the word facilitator and professional facilitator comes up very early. And you would naturally find such a person and ask them to come and help you with your meeting, and they would teach you and your audience a new way to think, and you'd put up a whole lot of post-it notes on the board and stuff, and at the end of about 2.5 hours, with this professional facilitator teaching you a new way to think, you would have the impression that you had done something really wise with your consensus generation, having been led by somebody who knows absolutely nothing about the subject that you were generating consensus on. So we have never done that. From the beginning, I've been the guy, or somebody like me, who is leading the consensus generation. And so I actually know something about the subject that we're generating consensus on. And so that's entirely different from what all the books say a way to generate consensus. And the reason that this works, you see, if people were going to die if we got this wrong, if like that, you know, if it's a life and death situation, if, you know, armies are fighting each other and stuff, you couldn't do consensus generation like this. But it's because, like they say in academic life, the stakes are so low, that you can actually be fair. You can have somebody who knows the subject and who is flexible, who's willing to change his general concepts based on new ideas generated in the room. So that's what we have done this whole period of time. And that's how the consensus has been generated. Until 2013, we had no structure whatsoever except the benign guiding hand of Dr. Lorraine Rackison and me. And then in 2013, people got the idea that maybe some structure would be good. So, you know, the Swiss have a good reputation for doing things right. So we created a non-profit Swiss foundation, the Banff Foundation for Allograft Pathology, which I chair. I'm in my second 3-year term. So in 2019, I will no longer be leading this, and Michael Mingle, who you may know, will be taking over from me. Here are some pictures from the meeting. And, you know, some students sort of begin in this course and then keep on doing things with me sort of forever afterwards. And if you end up doing that, you might actually end up in Barcelona in March of 2017, which is when the next Banff transplant pathology meeting is. But that's transplant pathology, right? So that's the pathology of an area of medicine that I told you was meeting the needs of only 10% of the people. So now if we think about regenerative medicine, you could imagine something 10, maybe 15 times larger than the entire field of transplantation, the entire field of transplantation pathology, which would be regenerative medicine pathology. And so that phrase, regenerative medicine pathology, in other words, what's wrong with stem cell generated organs, is not in any book or any paper yet, but we plan that very soon it will be. So the world is changing rapidly, and there's always resistance to change. People vary in how much they like this idea of jumpstart, because it's like jumpstarting them and maybe doesn't seem very pleasant, but in a sense we have managed to do that. And the transplant pathology people, not only pathologists, but clinicians, surgeons, basic scientists, people from pharma, from regulatory agencies, and so on, have all come to accept what I have been pushing since 2011, which is that transplant pathology is going to morph to regenerative medicine pathology, and it will be much, much bigger and much, much more important and more successful as a medical therapy than transplantation is today, when we have the possibility of rejection of organs. Presumably, when we're making organs from stem cells, they will not reject. So there are a lot of amusing coming together of different sectors of human beings. Yes?
Can you go back to the last slide?
I can, yep.
What are those slivers of red going up to what looks like a blood vessel?
That's blood vessel, right? And then, so as a part of granulation tissue in wound healing, new capillaries form, and in stem cell generated organs you have to have a supply of blood to keep the cells going. So it can never be just putting in cells to do stuff. You also have to be supplying them with blood, and there are some settings now where, for instance, you can make a stem cell generated liver and it'll work for a few minutes and then it clots like crazy, and it could have gone for hours and days and months and weeks if only you'd been able to keep the blood going, but you weren't. So the vascular supply is quite important. It seems now like it's hard to repavement more than about 80% of the blood vessels, and the remaining 20% have this incredible propensity to clot. And so that's one of the challenges. It's a solvable problem, but it is one problem. Yeah, so all those things need to be understood. Cell attachment, proliferation, migration, differentiation, remodeling, and providing a working blood supply. It's not only the concept that regenerative medicine created organs might work worse than regular ones, but also that they might work better, and some people might like that. So you might like your own heart fine, but you might like to get a second heart. So when you're running really fast, you don't get as winded as any of your colleagues, and you're sort of this super athlete. There are other things like that are part of human enhancement. So the improvement in health brought about by stem cell generated organs could go way beyond normal health to sort of, superpowers of whatever sort you might be personally interested in. And as I said in my first lecture, eventually medicine will be largely about that. When we cure most of the important diseases of today, and there's the question of whether physicians are morally ready for this next step, right? So there are enough moral quandaries just doing what physicians do today, but if we get rid of most existing diseases, and if most questions are types of enhancement, who should get them, what they should be, you know. I think that could go on for a very long time and generates very interesting questions. So regenerative medicine brings together like physicists and biologists and physicians and a whole bunch of different groups of people. And sometimes you end up with papers about things you just can't believe anyone would publish a paper on this. So this was a situation where they're looking at how to tell whether discarded human kidneys are a suitable scaffold for reseeding them with stem cells and stuff. So they seemed really, really perplexed by the fact that they couldn't see any cells there, but there was still DNA from the person who had owned that organ before. And of course, that's what happens. I mean, when cells die, the nucleus disintegrates, but it doesn't mean that the DNA is entirely gone. You just can't see it anymore. It's not a stainable structure anymore. And so this is basically like a whole paper about what happens if you just let an organ rot. Like if you take a living organ, put it out somewhere and let it rot, and then study it and write a paper about it. But this next article will be of interest and is quite valid, and that is, do Canadians want this? Do Canadians favor regenerative medicine? And in general, Canadian citizens are in support of longevity, radical life extension resulting from advances in regenerative medicine, and probably on average are more supportive than the more combative and crabbier US public, although I'm not entirely sure how the two compare. So I've talked before about the technological singularity, and when you apply it here, you can imagine that when we really are able to take an organ that used to belong to somebody else, flush out all the cells, leaving only the collagen framework behind, put in cells that are identical genetically to your cells, and then have a normal organ created and then put that in you, that's really going to be a game changer in just exactly the same way as machines being way smarter than people will be. So it's a different kind of technological singularity, but it's still equally important. And just to give you the idea, what are those three hearts down there doing? So the one on the left is a normal heart, and then you flush all its cells out and you get this ghost heart, right? The white heart. And then when the new stem cells you put in start to grow, it's pink. So that's not going to be on an exam, but I think many people would expect that you might know that after today. Yes?
How do you flush those cells out?
It's a combination of enzymes that break the intracellular junctions and stuff, and pressure.
And that doesn't harm the collagen?
Collagen's pretty tough. I mean, it's not that it's unharmed, but it is intact enough that new cells are able to grow on it. Yeah, it's kind of a science about not being too rough and still getting rid of most of the cells. Yeah, kind of compromise. So do things ever go wrong? Well, yeah. So like in this experimental model in rodents of stem cell generated kidneys, so they flush out all the cells and then infuse neonatal stem cells, so very famous publication by Song et al. And then you can stain for what cells are where, and like there are cells called podocytes that are intended to just be on the filtering apparatus of the kidney called the glomeruli. It sort of sits on the outside, and those podocytes, which in a normal kidney are never present anywhere else in the kidney except where the filter is. In this kidney, they're sort of wandering all over the place, and we have no idea what the consequence of that is going to be. I mean, we assume most of this chaos will lead to organs that function less well, but there'll be occasions, I think, where people find that actually things work better in this chaotic atmosphere of cells not. See, this is the thing, like physicists believe, since they know very little biology and medicine, that the homing of cells to know exactly where to go is like perfect. It's like magic. And because they're physicists and not biologists, they don't have to understand how the magic works. They just know that it's magic. And it would be easy to talk about if that was entirely true or false, but it's sort of like in between. Like cells are moderately good at finding out where they're supposed to go and attaching right side up and doing the right stuff, but it's not guaranteed, and they don't seem to mind also going out other places where similar cells have never been before, and so on. So, there are many problems with stem cell generated organs that are not being discussed. And so you may wonder like whether this lecture today is going to influence your personal behavior at all, or just going to be another one of those lectures that, you know, is a little blip, and, you know, for the next few hours, you know, a little bit about stem cells, and then very quickly the knowledge will completely evaporate from your brain and it'll be completely like you were before, right? But there's a possibility that you might be actually taken in by this slide that suggests that you could find a role for yourself here. There are many, many questions that really nobody's much looking at, and these are just some of them. The recellularized organ clots like crazy, impossible to regenerate more than 80% of the endothelial surface, it seems. It's hard to get the right cells in the right places. Many of the important cells seem to be terminally differentiated cells. Like, you know, you'd like to think that stem cells can create everything. It seems like some of the important cells, it's a little bit hard to get stem cells to make them. So, kidney progenitor stem cells are hard to identify, and for that reason, kidney work has lagged behind. And it's easy to make stem cell generated kidneys that lack the loop of Henle. Now, I'm sure the loop of Henle sounds to you like completely boring, but without the loop of Henle, the long loops that go down into the medullary interstitium, very hypertonic and so on, you may wonder how you're able to concentrate your urine, or maybe you never wondered that and really could care less, but it has to do with these long loops of Henle, without which you would not be able to make a urine that is more hypertonic, more concentrated than normal saline. And so, because it's so easy to make kidneys that lack long loops of Henle, there are two sides of this. One side is a group suggesting that you put in these kidneys lacking the long loops of Henle, and you just pee out your body water, all of it, over about an hour and a half, and you're dead, right? This would not be a good thing. So that this would create a lethal polyuria. And then there are other stem cell biologists who believe that they actually think that won't happen, that they can figure out a way that a kidney without long loops of Henle could still function in a way that would benefit the patient, and they may turn out to be right. But Those kidneys lacking long loops of Henle certainly will not be functioning normally. They may function in a way that's okay and benefits the patient. And the other thing that's intriguing, like for those of you who took physiology at some point and may think that this knowledge that was like wasted time in your life, thinking of all the things you could have been doing that would have been a better use of your time than. So there are many of old-fashioned concepts of physiology that are new and exciting again when applied to stem cell generated organs, because you have all sorts of things nobody ever anticipated, like podocytes wandering around in the other parts of the kidney where they're not supposed to be, and a bunch of things like that. So how normal does a kidney or does some other organ need to be in order to provide useful function for the patient? That's going to be an important question in tissue engineering pathology. So transplant pathologists will become tissue engineering pathologists. It's not something beyond us and not something beyond you. You might be able to find an area, and the idea that you can use your nostalgia, you know, those old ideas that you thought you had been forced to learn but are completely worthless in 2016 turn out to be pretty useful. So beginning at the beginning, I've been trying to write about this. If you start something brand new like tissue engineering pathology, how do you conceptualize writing something or beginning something that's brand new? There's a lot of exciting words been written about beginning at the very beginning and blah, blah, blah. But do you know London's Crystal Palace? So this is being rebuilt, I think, in 2017, 2018. It was the largest glass-walled building in the world when it was made originally in 1851. And in 1851, to celebrate the opening of the largest glass-walled building in the world, they had this grand exhibition of technology, and that included the International Classification of Diseases. And you may know that this is something very important in medicine, and like, you know, if you're going to treat diseases, you have to have some sort of classification of diseases. But from the beginning, the emphasis has been on death. Death, 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 nothing but death. So cause of death and how many deaths and so on. So the classification I plan, like the Banff classification of tissue engineering pathology, will be about sustaining life and not very much about death. So some of the interesting things, you get a lot of variation in terms of what the kidney or any other organ looks like under the microscope. And so you might say, can you really call this a kidney? And I think the answer is yes. So if it's in a kidney scaffold with kidney cells and so on, no matter how you look at it, it's a kidney. It may be a kidney that won't work, but it is a kidney. So the idea is to describe what might be wrong and whether it will work. So, some of you may know Corey Fung. He was a student in this class 3 years ago. And he's the only student I've had thus far who can draw. That doesn't mean none of the current students, but I haven't tested them all. But anyway, so he's done these drawings of where the various abnormal states that you might encounter in tissue engineering pathology might be. And so, like, is there too much endothelial disruption? What's the chances of tumors forming? Whole bunch of stuff like that. So, I'm going to stop at this point. This is a picture of the mule deer, which are present great quantity around the Banff Center, and that is the Trans-Canada Pipeline Pavilion at the Banff Center for Conferences, where the first Banff classification meeting was. So we've come a long way, and tissue engineering, regenerative medicine is the future, and I'd be happy to answer any questions you may have about this. Yes?
How do you test the function of a synthesized kidney?
Yeah, so I mean, there are many sort of knee-jerk reaction answers to that. So, like, if somebody were to test your kidney function, they'd be basically thinking about glomerular filtration rate, GFR. So you could determine that. When you look at serum creatinine, that's a sort of ballpark way to look at that. But there are many other things that the kidney does. It produces renal erythropoietic factor, which causes red cells to be generated, without which you don't make new blood cells, you know, a bunch of other things like that. So it's not just filtering waste, it's also doing a whole bunch of other things. And that's true of all the other organs as well. You know, the liver, I don't know how many functions the liver has, probably several hundred different identifiable functions, you know. Like, it's interesting that the thyroid, in animal models, like stem cell generated thyroid is easy to make, and you can do a lot of exciting things. But generally, thyroid function can be replaced with a pill, a single pill a day in the hands of most people. So you have to be a bit careful about getting too excited about stem cell generated organs in a situation where, you know, it actually, if you can do the same thing with a pill, that already exists. But for most organs, that isn't true. It's just the thyroid, which is to some extent replaceable with a pill. Yes, Earl?
Well, I have a couple of questions for you. The first one is, has everybody in the Banff group bought into this transition to regenerative medicine?
No, I wouldn't say so, but what excites me is that the young people have. In that is the seeds of, you know, you can tell what the future. Yeah, so I gave a talk about this, and a number of like youthful leaders told me that the other young people in the audience really liked this talk that I had given. So that gives one faith that eventually they'll all buy in, you know, because like some of the guys who are digging in their heels and will never pay any attention to this will die off, and, you know, eventually. Right? So, yeah. Yes?
Since I don't know too much about how they actually build new organs, maybe my question will be way off base, but I'll say it anyways.
Yeah.
What's the sort of, how advanced and how many different ways do they have of making those organs? Because you showed the like the collagen matrix that they just seed the cells on, and then I assume they can also just seed cells onto like a platform that has cell food on it.
Right.
And then try to grow themselves. Like, how many different ways do they have of trying things at the moment?
Yeah, well, the simplest way is to use a decellularized matrix. Take like a heart and flesh out all the cells so you have only the collagen, and then flesh in new cells. Depending on what the organ is, random arrangement of the cells may work reasonably well. Like in the liver, I'm not a liver person, so this may be like an anti-liver bias, But I kind of have a feeling if you just mixed up a bunch of liver cells together and got them to grow happily in a random way, they would do quite a bit of good for the patient. Kidney cells, that's not true at all, because the parts of the tubule need to be like lined up, and you need to get the sequence, proximal, distal, collecting, all kind of thing. So if you just randomly mixed a bunch of kidney cells and go for it, guys, whatever you want to do, that you just get a mess of cells with a high, metabolic rate that require a lot of blood flow and oxygen to stay alive, and except for producing renal erythropoietic factor, I guess they probably wouldn't be doing anything useful. So the question whether human beings know enough about what the actual sort of 3D and 4D, if you want to think about it that way, how things are supposed to change over time in the normal organs, that without using an existing scaffold, whether we can start from scratch and create a useful organ. I think at some point we'll be able to do that, but in most organs we probably don't understand how they work well enough to do it from scratch. Yeah, I don't know if that answers your...
My follow-up question to that, because I did my first degree in genetics, I know a considerable amount of the way organs form has to do with factors outside of the organ itself.
Right.
So it has to do with like the tissue around it excreting specific factors that tell it like which way is up.
Yes.
Back, left, right, all that kind of stuff.
Right.
How was that done in transplant?
Yeah.
How did they do that?
See, I think that this is a very exciting area. Embryogenesis does kind of seem magical to me. How do those cells know exactly what to do and when and where and so. But it occurs over a particular time frame, and in a womb, it's been developed over thousands of years, so on. So in a stem cell generated organ, many of the same processes take place, but not as well because of the same sort of thing that you're talking about.
So they put it in the scaffold, or they put it on the surface that they're growing it on? Will they just put those?
They put in the known growth factors and that sort of thing, stimulating factors that have been shown to benefit that kind of organ culture. But they're probably missing out a lot of important things that they don't know about yet. So I think, that...
But there's no sophisticated way of doing that at the moment. It's just like the ones we know about, we put it on the surface, and then we put the cells on the surface.
Yes, I think that will obviously change with time, but that is the case now, yes.
So Kim, I have a fair amount of skepticism about this, as you know.
Yes, I know.
And my skepticism is also fed by the new knowledge that the stomach produces about 80% of the serotonin in the brain, which means to me that we have an integral system, and replacing one system may not put that system into the other systems in the body.
Yes.
And so a regenerative model has to include the whole kit and caboodle in order to make it work. So I'm wondering if, like, emphasizing a kidney or a heart or, an individual organ is really going to get us where we want to go.
Well, I know this isn't where you intended to go, but what I can say about that is, the non-biological substrate, and when human being consciousnesses become comfortable having that. So once you're non-biological, you don't need oxygen, you can travel much faster to distant places, there's so many limitations that we'll have forever as long as we are biological. And so, yeah, I sort of see it as a stopgap. I know it sounds crazy to say that, but you know, we'll eventually get beyond feeling that, you know, we have to have a heart, and you know, that all these organs are necessary for us to be who we are. We'll come to accept that we can be who we are with just our mental activity and being able to act upon the world in all the ways that we would like to, but that does not include necessarily having flesh and blood at all. Yeah. So I'm sorry, maybe it seems to be leapfrogging a whole bunch of, you know, intermediate stuff.
I mean, that's several hundred years according to my calculation in the future.
Yeah. Yeah. Well, it's interesting to speculate, like the people in their 20s, what can we guarantee that we talk about now will actually occur for them, you know, in the central part of their lives. And I think you know, regenerative medicine certainly will. Uploading our consciousness to non-biological substrates is probably beyond the lifespan of the people in this room, but I don't know that for sure. Yeah. So anyway, that would be my thought. Any other questions? Thanks for being so patient with this. This, of course, it's a kind of talk that I give to many different audiences. I didn't think before I gave this today that you'd all been to medical school and, you know, so on and so forth. So the parts of this that assume that you knew things that you don't actually know, you don't have to worry about. So. Okay, any other questions? All right, we'll see you next week. So next week, so Pokemon Go, if those of you have friends who are really into that, you can ask them to come on Tuesday. And then Jonathan White's Ask Me Anything. It doesn't work very well if none of you have watched any of his videos. It's sort of intriguing that, as I've said, he has held all sorts of different opinions. If you look at, you know, the Gartner hype cycle, he's been at every point. So I hope you come armed on next Thursday week from today with really good questions. But even if you don't, it'll be an outstanding session. So something to look forward to. Okay, great, thanks.